Good morning, everybody, and thank you very much for joining our SPHN webinar. We have a special guest speaker today, Michael Baudis, who is presenting a project that runs actually outside of SPHN on a more global scale, but is partly supported through funds of the Biomed IT project. Michael is a professor of bioinformatics at the Institute of Molecular Life Sciences of the University of Zurich and a group leader at the SIB. He's actually a medical doctor by training, but more of a bioinformatician in his current life. So his group, his computational oncogenomics group, is centered on the comparative analysis of genomic profiling data in cancer with a focus on somatic copy number variations. Apart from the analysis of cancer genome data, in recent years, Michael has been involved in efforts to facilitate the access to genomic data for research on an international scale. Since the foundation of the Global Alliance for Genomics and Health, GA4GH, in 2014, Michael has been a member of it and currently serves as the steering committee and as a co-lead of its discovery workstream. Related to the work in the Global Alliance, Michael also co-chairs the ELIXIR Beacon project, which develops APIs to query genomic data collections in a privacy protecting manner. And this is actually the project that we very much look forward to hearing more about today. So Michael, passing the word to you. Thank you very much for joining us today and taking the time for this webinar.
Thank you, Katrin. It's always a pleasure to have the opportunity to do something with SPHN, because this is pretty much in the center of the work I'm interested in, to facilitate this sharing and interaction around genomic data. So the talk today will give you first a bit of an introduction around the background, GA4GH, the Beacon project, and then some applications, and especially where this will lead to in the future and what future we foresee for the Beacon project. So as Katrin, this is just a brief introduction for people who don't know me. I saw some of the list who I'm very familiar with. My background, as Katrin said, is basically I'm a medical doctor. I studied in Heidelberg and worked in hematology, oncology, and later then drifted off towards research, first really lab research and at the GAUG and at Stanford and University of Florida, and more already realizing that not only the experiments are important, but the generated data from these experiments should also be accessible and should also be aggregated to make larger use of this. And this basically comes to something which I wrote. Basically, it's a thread going through all the work I'm doing in format development and also in my own resources and research. University of Zurich currently, and as Katrin said, very involved in Katrin said, very involved in these Global Alliance and ELIXIR project. Let's go. So yeah, so this is something people involved with SPHN are obviously very familiar with. I mean, genome screening is at the core of what we call now personalized health. Genome analysis have become cheaper, have become more ubiquitous in clinical practice and in research, although, as also most of you are aware, the genome analysis we as researchers want to have, that is whole genome and complete genome screening and complete data availability. This is something which doesn't always intersect what's happening in the hospitals, but there is whole genome and exome technologies also become cheaper, more ubiquitous, and hopefully will find also their way more towards the clinic, and so that this data can then be collected. This is also due to the large amount of sequence variants in the human genome, which cannot just be covered by panel sequencing efforts, although those frequently give the best value for knowledge or knowledge for money in clinical setting. So important for getting towards the data, which is produced in lots of different places and lots of different institutions and clinics and research centers. And there's a map that shows some overview of worldwide genome screening activities focused on sequencing and copy number duration, is that we are able to exchange the data. And this is what we are working on, making data available and also making the protocols for these. As an overview by Heidi Rehm, who now leads the genome efforts at the Broad Institute and is one of the co-chairs of the Global Alliance for Genomics and Health. And this basically gives you some ballpark numbers of how the numbers are developing and what the targets are of these genome analysis, which start to accrue more and more in clinical and research settings. So rare diseases are obvious target, but cancer will become more and more a target because just of the high incidences and the high applicability, and also the opportunity to understand biological trajectories there over time in diseases with multiple analysis and so on. So we are going from the thousands to many millions throughout this decade, and that's an extrapolation, obviously. But this data sits, as I mentioned before, in many different places. And so there is no way, there's not a single database to rule them all. There are annotation databases, but even these annotation databases cannot cover everything that's coming in, all the knowledge that's being generated. So there has to be a way to access data in multiple resources. Data has to reside in multiple different places, but one has to be able to make use of this. And this is basically the principle of federated data access and analysis over federated data networks is one of the founding reasons of the Global Alliance for Genomics and Health. The GA4GH, making genome data accessible for research and health, could be seen as the paradigm. And it was founded based on ideas, some brainstorming in 2013. And GA4GH was founded with some events in Kingston and London in 2014, and has then with yearly plenary meetings, for instance, the 2018 was in Basel, some of you may have attended this, is steadily growing and expanding, and really especially expanding internationally with lots of growing number of participants. Recent plenary also big contributions from Africa, from Australia, from Japan, and so on. So it's really becoming more and more a global effort. That's just a bit of a timeline. And the idea behind GA4GH is that, as the white paper, which is listed there, mentions, to create a federated ecosystem for sharing of genomic and clinical data. Clinical data here is not something like access to full electronic health records, but it is access to standardized, deeply annotated, consistently annotated clinical data like phenotypic data, like some progression markers and so on, which can be captured without really sharing boatloads of documents here. And the way this is being done, you can see here, or this is envisioned, is basically to provide different frameworks to facilitate these data sharing and data federation activities. This can be through direct APIs, that is data access layers to directly access databases and queries over the internet and so on, but also through providing guidelines for how such resources can be set up and how the data should be handled on an ethical basis, on a security basis, privacy basis, and so on. And this one of the most successful, and this is what I will be talking mostly about, is one of the most successful projects here has been the Beacon project, which is one of those which should facilitate end users to use an API to access different resources. That's a very wordy slide, and you may look this up later, but actually I show this here because I just was digging this up a while ago again, and then I recalled this meeting in 2014, and that basically many of the foundations, when we talk today about these Beacon protocol, about genome beacons and so on, they were all these foundations, and these lots of details were actually already described by by Jim Ostel at the meeting in 2013. So there are some keywords which are very important. It should be an international effort. It should look for signs of willing participants, which want to contribute to that, but may not even directly know what the roadblocks, what the issues are in developing such a data sharing protocol. However, also this project from the initiation was thought to not just being a game project, gaming possibilities out, but really to provide measurable signal and so really provide some functionality directly from its first instantiation. And the functionality, the expanding of functionality, especially here, frequency of layers, regions, and so on, more complex queries and so on, those were thought of being something for a later issue. And actually he mentions specifically version 2. I will come to that. So what's a beacon? A beacon is, well, it's basically an API. So it's a piece of software which answers a query for a certain genomic variant query formatted in a certain way. Here you would see, is on chromosome 17 at this position, has the G be replaced with an A, basically in the sequence. Okay, so a beacon answers a query for a specific genome variant against individual or aggregate genome collections. So it doesn't have to be sample-specific collections, but it could be aggregate collections where just frequencies for such alleles are stored. And then you see, yes, this variant is there or it's not there, and so on. And this doesn't seem very informative, but actually it becomes informative if you do this, and that was the beacon protocol being made for, if you can do this over a network. So if you have now the possibility to say, oh, same query, but you send it out to resource providers worldwide, and then get a response back, is there a variant in this database, this variant, or isn't it? And from that, you can learn already something. For instance, if you have a candidate allele which may be related to a rare disease, you may find out where other patients of the rare disease are. Or if all the beacons respond with, yes, we have these alleles, then it's probably not something disease relevant, but maybe something just part of the genomic background, right? In this example here, we would say, oh, there are some red arrows. We have found this variant in some resources, and then one may know that they are associated to a certain disease. So there is some real information content, but it's very limited. The protocol, however, was taken up enthusiastically. So in 2016, there was a beacon network that was developed by DNA Stack in Toronto, and already tens of organizations, hundreds of data sets and so on, were part of this network. So you were able basically already then to send these simple queries for these SNPs and get responses then from a large number of resource providers, which could be big institutions or just small research centers with single data sets and so on. So, and this was then be aggregated over a network. So there was uptake. This was the intention from the beginning, and there was some practical use, but again, still limited. Now, in 2016, basically, ELIXIR came in and took development of the beacon protocol under its umbrella. So ELIXIR, for people who don't know, this is the European Bioinformatics Infrastructure Organization, to which most of the bioinformatics organizations in individual countries in Europe and a bit beyond, like Israel and so, belong. In Switzerland, the local node is Swiss Institute of Bioinformatics, obviously, right? And ELIXIR became a direct partner of GA4GH because they have very overlapping core messages for a part. So, and this would be to simplify the way people search and request access to potentially to data in international and national genomic data resources. Directly, one of the missions of ELIXIR was that these beacons, which had shown their basic utility and their wide adoption, should really be pushed towards becoming more biomedical for research and clinical application. So, and some issues were originally identified and then divided between the participating groups. For instance, authentication, then the identification which bioparameters besides genome data should be able to be answered for maximum functionality. Clinical variant formats, not everything is a SNP in genome variants. So it should be found in integration of beacons into secure local environments. So how do you integrate authentication with the beacon protocol, or for instance, like incidence of local EGA? Also, it was clear then that we needed formats for data delivery, and data delivery is a tricky issue because one of the advantages of beacon protocol is with the yes/no answer, it basically doesn't expose information about the patients, about the individuals, which are being part of these beacon collections, but you need a data delivery, so there have to be ways around it. And how do you interact with electronic health record standards? So the beacon protocol was, the development of the beacon protocol became part of the, or focus of the ELIXIR Beacon project. So one has to, it's sometimes a bit of tricky to figure this out. So we have an GA4GH beacon standard, but then we have the ELIXIR Beacon project, which develops the beacon standard, but the standard is not owned by ELIXIR itself. And lots of people from outside of ELIXIR contribute to the development. The beacon, official GA4GH beacon standard actually was already then the version 1 being developed by ELIXIR, and we are currently on version 1.1. And this already, and this has came into effect in already 2018 and 1.1, I think. And already from an earlier version, from version 1.0, it provided some support for features beyond the original pre-ELIXIR Beacon project protocols. And for instance, these were support for structural variant duplications, deletions, and so in beacon queries, and additional structural variants in principle being possible. The version 1.1 already included a data handover protocol, which allows data delivery from beacon queries, but outside of the beacon protocol. I will talk about this. The ELIXIR AAI was implemented. It's an open authentication system. And already there was made some groundwork for version 2 features like filters and extended quantitative responses. ELIXIR beacons now exist, which are more closer controlled than the just open implementations of the protocol. Exist now in several ELIXIR nodes, and here is a list from the middle of this year. And these support the new protocol. So the old ELIXIR, the old beacon network doesn't support all the features of the current protocol, one has to say, but the ELIXIR network does. And also ELIXIR network goes now beyond, and with the support here for authentication systems, so allowing secure environments, and in a layered access way, saying you have different levels of access. You can provide public access, registered access, or controlled access. This is then a screenshot here of the ELIXIR Beacon network entry with some of its nodes. This is, as things are done in ELIXIR, it's an effort through many different nodes. For instance, the network here is maintained by ELIXIR Finland, but the authentication is by ELIXIR of Czech Republic, for instance, provided. So you can now authenticate with ELIXIR AAI or other, or this is linked to other AAI providers, for instance. Switch would also work there as a provider. There are now incremental extensions of the network, adding more and more nodes, which have to fulfill all these criteria. And for getting more nodes in, there is a registration service where one can upload data, basically, and self-register to the service. And this, the protocol, but also the network, serves as an integrator throughout ELIXIR human data efforts, and also is a starting point to go beyond ELIXIR with getting other interested parties in. I mentioned the handover protocol as an opportunity to deliver data through beacons already, and I will show this later. But what this basically does is you have your standard beacon query or additional parameters now coming up in version 2, and then your backend delivers a beacon response, but then it delivers also handover objects, which are basically URLs pointing to some way of data delivery. And this can then go through an additional authentication and provide the data through these different handover action. And this could be phenopackage data, graphics, variant data, and so on, which I will show. This is basically how this looks just as part of the API response. And here I show some from our own implementations. So these beacon handover objects do not contain any data, but they just contain pointers to data, right? And these pointers have to be resolved to the resource, but they, in principle, cannot identify, for instance, individuals in the data collections as by themselves because they're just anonymous objects. And there is, in principle, no need for separate endpoints, although we will come to this later with version 2 development. So you have a very easy, flexible development through these opportunities. So what's currently possible in Beacon? Beyond the willingness, testing the willingness for data sharing, as originally precise queries. We have now range queries, so you can basically specify a range from here to there and various variants, not only precisely specified variants. Already the responses deliver frequencies, structural variants like deletions, I will show this, and any data matching a query, variant, sample information, patient data, and so can in principle already with the current protocol be delivered, but as a sideline to using this handover project and AI integration. So it's already something very powerful, which can be used. However, how can this be extended further? So the important part is to engage many different stakeholders and partner communities which already deploy or which are targets for deploying Beacon. And here is a map showing you basically the logos of many of these institutions like EBI, EJPRD, SIB, SPHN nodes, and so on, the Human Copy Number Variation Project, GEM Japan, and CRG, and Genomics England, and many others. Yeah. So, and these are all, many of those are we are talking with when developing now the protocol further on, and I will come to this next. Beacon version 2. Beacon version 2 is something which has been in development now since basically sometime last year. So, and this is a radical reworking of the Beacon protocol. So instead of, for instance, having just a single type of, it's thought as a very modular protocol where you can add more and more features over time. So instead, for instance, of having just a single type of variant query, which can be very complex when querying for different types of genome variants, there will be scope variance queries. There will be access levels to allow you to have greater data access as shown before with this open or registered or authenticated. A core concept is filters, which I will present more in the next slides. And other types of queries are also being evaluated and maybe made possible, for instance, in clinic context. How can you directly query for certain patients and so on and retrieve their data and so on. Importantly, since this will lead to a large heterogeneity in Beacon endpoints, schema versions and service infos will be provided using standard GA4GH protocols so that the different endpoints can actually tell you what queries they are able to answer and what data they are able to deliver. And this is already, some of this is already in rollout. This is a draft white paper which was finished recently. It just gives you, this is just the introduction, but it shows you some of the evaluation process the current development went through, especially with the clinical, to fulfill the clinical concept from looking at data flows in clinical genomic analysis and precision medicine towards developing data models which address specific points which hadn't been addressed in the Beacon version 1. There was, for instance, a bit of confusion if you talk about aggregated variant data, annotated variant data, variant evidence, or if you talk about sample variants, that is single observation, for instance, in a patient. And lots of stakeholders were involved here: BRCA Exchange, Autism Speaks, GEM Japan, SWIP, and so on. Now, this is what, in principle, for the now thinking about how a Beacon V2 network, not about single instances, but how a network could look like instead of the simple variant query we had before. It should be, one should be able to do something like that. Here, for instance, you have a query where the query parameters are shown here, and you already see there are lots of strange, for people who don't really know this, strange ID-like elements. And these are queries here, NCIT Duo HP, for instance. And basically what this query here does is it asks, have you seen deletions in this region on chromosome 9? And this would be a tumor suppressor gene locus from a juvenile patient, HP, in a data set with unrestricted access. The Duo code here says it's unrestricted access. NCIT says it's a glioblastoma. HP is a code for a young patient, right? And this can then go through this authenticated network with or without authentication. And then lots of different types of data can be delivered. I showed this for some strange query concept. So this code NCIT colon and C and number, this basically is a code, as I said, for tumor type. And here we learn more and more, and that's a general move in data science, that we go more and more to these hierarchical or ontology systems for data annotation. And this is, for instance, here, cancer ontology, NCIT, shown as a diagnostic ontology, which is much more powerful, especially for these queries, because you can query on different levels of the ontology in Beacon using these codes as so-called filters. But there are many other ontologies and services like OXO and so on, which also help you to disambiguate such things. This is the, for instance, implementation of NCIT Neoplasm Core in our resources, where you are basically able to query here now for skin carcinomas or cutaneous lymphoid neoplasms. And since you used an ontology, it then derives all the subclassifications of these, so you don't have to provide those, but it will directly then use this tree of classifications and query for all of them and delivers then a response. So, and these can be used as this Beacon filter concept. There are other parts of filter concept which we develop, like fuzzy or specific annotations. Important is that data in resources is annotated in standardized way using these kind of codes, so that which facilitate and only allow these queries. And also this actually is then very powerful, because the Beacon resources can then also provide the list of supported systems, so queries can be formatted in a working way. And we use their other formats, standard formats like ISO codes and so on, and Mondo Monarch initiative derived coding systems. Now, if one has these different codes, but also data schemas, it's a bit hard to get the overview. So GA4GH is developing many different products and protocols, projects, schemas, and so on, but some of them are deeply integrated. For instance, the future or current default data transmission format for variant and phenotype data is now becoming phenopackets, but your protocol may not use the whole phenopacket implementation, but may just use the same way, for instance, to annotate age or phenotypes as Phenopackets does. So we developed in GA4GH the Schema Blocks initiative a while ago, and this is also supported through SPHN, which is a cross-workstream, cross-driver initiative to provide object standards and prototypes, vocabulary and semantics, and which is kind of the exchange place where different protocol developments go to pick or deliver small parts of themselves or of other schemas to reuse these other schemas, so that one has consistent language structures and implementations through different protocols used, for instance, always the same way to annotate a prefix ontology, either for reporting or for querying, or to use the same time standards for age and so on. This is all important. These are, for instance, examples here how these protocol parts are shown here. And I mentioned before, Phenopackets is now a big data delivery and exchange standard. However, one can take parts of it out and represent them and reuse them in different areas. For instance, how do you reference a certain genomic file format, or how do you reference a biosample and what should be the attributes in a biosample and so on. So this facilitates the normalization. And from this, I will come briefly towards how this is being used in test implementation. So how we drive now Beacon development through our own resources, but also other people involved now make use actually of the features of current and already upcoming Beacon version in practical implementations. And I just want to briefly mention from the Center of Genome Regulation in Barcelona, the COVID-19 Beacon they launched this year, which is now something very new, not a human variant repository, but something related to COVID, and then also talking a bit about our progenetics resource. And this is just an example how a beacon can look like, which is now not even being focused to humans. So the CRG people, they are also involved indirectly or driving the Beacon protocol development. They created an information resource where basically one can search and scroll and identify genomic variations in COVID genomes, not in human genomes now, but then also can relate to the provided information from host information of the humans which had been infected with this disease, with this virus, and already can gain a lot of information about which domains are involved and so on, all through this resource, which basically runs on a Beacon framework. Our own progenetics database is a copy number variant focused genome variation database in cancer, which more than 130,000 samples now, and actually the whole front end runs on a only slightly modified version of the Beacon protocol. So we use this implementation actually to drive the development, to really look how can we make our data structures better and what is needed as future additions to the Beacon protocol to enable functionality like we have here now. So the consistent use of hierarchical diagnostic code, as I had shown you before, allows you to use the Beacon filters, and we just do filter queries if you want to have these disease code, for instance. And if you see here this data shown here, these plots, and so they are all basically accessed using Beacon handover protocols. That's a principle how a Beacon front end can look like. Now here with example for structural variant, as I mentioned before, deletion in a certain gene. Here again, CDKN2A and glioblastomas, using NCIT code here as a filter, have as positional variant query, some visualization of this, and then querying the database, and then actually using handover object, which just points to the identified call sets to generate histogram, for instance, here for copy number variants, showing here, for instance, the deletion in this spot we searched for, but also then the associated other changes in this glioblastomas, nearly 700 samples matched here and so on. And then there are different export versions, and this is all driven by Beacon query and handover responses. For instance, here now you are able, if you do these queries on our websites, to then export the sample data and wrapped into phenopackets format, and this is how this would look like. Also, this handover can be used, since our resource is open, you don't have to go through any authentication step, but in principle can directly now go and download the variants now, which had been found, for instance, as in JSON format or in tabular format and so on. Again, all basically based on handover pointers provided through this. So this just to show you that one can really use already this protocol and use this as not only as an added value to a resource and to an interface you have already, but really to revert back and say, okay, let's rethink this, and can we make use of this protocol ourselves and develop a software stack directly around the Beacon protocol for a large resource. So nearly the last slide. Beacon, GA4GH, and SPHN, again, a wordy slide, but let's just go through the points, because I think this is going back a bit to the understanding of why are we doing it and what we are doing here. So GA4GH is, in my opinion, the major go-to organization for development of data exchange standards and implementation guidelines for genomics and health-related data with focus on biomedical research. But essentially, so GA4GH is a collaboration of volunteers and voluntary organization of contributors. So essential for its success are national partner organizations such as SPHN, and then inverse SPHN has then, and the other partner organizations, also ELIXIR, GEM Japan, and whoever contributes, they have the opportunity to shape the development of GA4GH through their contributions. But importantly, they also stay on top of developments and benefit through the alignment with international cutting-edge standard developments. So this is also helps to avoid waste in, for instance, protocol development and resource development. Early adoption of standards such as Beacon and phenopackets drives innovation and makes actually research easier for participating institutions, individuals, researchers, and also helps clinicians to really understand where these developments are going. And if you think about Beacon as some interface or added value for your local, whatever, variant database search in your clinical context, Beacon right now and a little bit in the future may still be limited compared to directly tailored developments, which then obviously require lots of development effort. You can put in legacy systems. However, if you implement protocols, open protocols such as Beacon, you already benefit from the possible integration with outside data resources, for instance, to get variant information directly integrated with your local EHR systems from the outside using the same protocols. And Beacon version 2 is specifically being developed with clinical requirements in mind. And it will cover a broad range of use cases in precision medicine, rare diseases, and cancer. And everybody is welcome to give input there. And the active participation of SPHN also supports a leading position of Swiss contributors of Swiss bioinformatics and biomedical researchers directly in the GA4GH development process. Now, again, this slide I'd shown already, Beacon v2 in an international network, doing queries which you can be interpolated in URLs and getting data delivered through internet protocols. And we can call this sometimes the Internet of Genomics. And we think if something like an Internet of Genomics develops, then Beacon will be at a core protocol in this endeavor. And the internet started small. This is there, as a reminder, the cover page of Tim Berners-Lee's information management proposal at CERN in 1989, and this is the first worldwide web page. And if you look and on the proposal, it was vague, but it was deemed vague but exciting by Bernoulli's supervisor then. So we still have a bit of way to go, and we hope we will come to also a more viable solution in the future for our Internet of Genomics. Here are some resources. There is a Beacon web page where you find some of this information of what's going on. Here is a link to our our Beacon installation on top of Progenetics, but actually you have directly the same search interface directly on the web page itself. So this is just showing that separate. And here is all the developments of software, and so are in the end mirrored on GitHub. So most of the things are accessible, sometimes with a bit of delay, but also our Beacon instance now has actually a GitHub repository, and you can actually get the code for a Python-based Beacon installation directly from our side. Thanks to participants, and obviously the list cannot cover everybody here. So my group at University of Zurich and an outside collaborator for the web development, Beacon API leads, especially Jordi Rambla is the one pushing the development forward in Barcelona, and Juha Törnros for the networks in Finland. My co-chair of the discovery workstream, Mark Fiume, had been the first developer of the original Beacon and of the original Beacon development, and people at Alexia and at GA48 especially. So thanks everybody. And the last slide is contact information, and the slides will be posted. So you can then look this up later, also the kind of early slides, reread them where it was too extensive. Thank you.